My name is Koichi. I'm from MD Anderson, and I am one of the early customers who had a chance to use this exciting platform, Tapestri. And we use this for about now 80 cases of acute myeloid leukemia cases, and I would like to show some of these data here. And other data are also going to be presented at 4 p.m. in room A302, so if you have time or if you have time to move, please go and listen to that talk too. So I probably shouldn't show this in the AACR meeting where cancer research is the focus, but this is the traditional kind of oversimplified cartoon of how cancer cells develop. But the normal cell acquire mutation A, B, and C, and then transform to malignant cells and create this monoclonal aggregate of the cells. However, we now know that this is really not true by a lot of next-gen sequencing studies that identify that cancer is aggregate or of more genetically diverse cell population, which we often call this clonal heterogeneity or sometimes intra-tumor heterogeneity. And this is really important, and because clonal heterogeneity is a reflection of how each clone evolved under the selective pressure of tissue ecosystem or under the therapeutic pressure from the cancer targeted therapies. So profiling clonal architecture in precision at any time points, like here or here, enable us to actually build the phylogeny of how the cancer cells evolved. So it is really important to characterize the clonal architecture of the cancer cells at the precision. And it also has a clinical implication too, because the WashU people nicely illustrated using the bulk sequencing method that identifying subclone at the time of pretreatment, which can constitute the relapse. So really precise characterization of clonal architecture also has some clinical implication too. And much of these work has been done by next-gen sequencing of the bulk tumor samples. And most of the times we create this ranking system of variant allele frequency of multiple mutations and then infer the clonal architecture model. However, this method has inherently limited because by just looking the VAF ranking, you can have multiple possible clonal models, and you never can precisely determine which model is correct unless you have longitudinal samples. So the single-cell DNA sequencing has a potential to address some of these issues, and our colleague Navin has been pioneering this, starting from the copy number profile of breast cancer cells and also single nucleotide variants of 50 breast cancer cells, both published in Nature here. And also Jerry Radich from Fred Hutch did perform genotyping of 2 genes in AML samples, roughly 500 to 1,000 cells. And just by looking 2 genes, you have this level of clonal heterogeneity that you can see in 6 AML patient samples. But historical method has some limitations too. When you do the single cell whole genome sequencing, the problem is the low cell throughput. You can only analyze about 50 or 100 cells, which is underpowered to really characterize a clonal relationship of multiple mutations. But if you choose a targeted approach like 1 or 2 genes, you may be able to interrogate more cells, but you are underestimating the real clonal heterogeneity. And also with a lot of whole genome amplification that is embedded in the protocol for a single cell whole genome sequencing, that creates a lot of artifacts and errors, which will also confound your analysis. So the Tapestri from the Mission Bio have solved some of these limitations. This was published in Genome Research last year. They do a targeted sequencing method, but not only 1 or 2 genes, but you can target about, here for this platform, we targeted 19 genes, but now I think with the latest platform, you can go to much higher numbers of genes and higher numbers of amplicons. And we were able to sequence about 4,000 to 5,000 cells. So this is a number that is much higher than what was possible from the previous methods. And we show that in this first pilot case where we had a diagnosis sample and relapse sample, we clearly saw the clonal heterogeneity at the time of beginning and also the remodeling of this clonal heterogeneity at the time of relapse. So since we were very happy with this pilot collaboration, we expanded this effort in larger scale sequencing data. Most of these work were done by two postdocs in my lab, Dr. Morita and also Dr. Wang, who performed the bioinformatic part. So far, we have finished about 70 AML patients being sequenced with this Tapestri platform. The initial AML panel, which targets about 19 recurrently mutated genes in AML, and also which targets the 40 amplicons of these 19 genes. And samples were either sequenced by the MiSeq 150 or 250 parent end. For the FLT3 ITD cases, we prefer to use this 250 parent end because it will cover most of the FLT3 ITD. And we use the GATK haplotype caller or ITDC combination to call the mutations. And we also did a concurrent bulk NGS as well as ddPCR to validate the single cell DNA-seq detected variants. And we are very happy to see that 100% of the mutation that was called by the bulk-seq was also called by the single cell DNA-seq platform. So there's a really good concordance between the bulk-seq and the single cell seq. And we did some SNP array data, which I'm not going to show today for the time, but we also did a SNP array to confirm the zygosity of some of these cases mutation. This is the clinical characteristics of these 70 AML patients that we profiled. We mainly focused on normal karyotype sample here because we wanted to avoid the allelic imbalance that will mislead us on the genotyping. Most of the samples were taken at the time of pre-treatment, and some of the samples were taken at the time of relapse and refractory time points. This is the QC metrics of these platforms. We normally input about median 270,000 cells into the machine, and we only get 3% of them being sequenced. However, this still results in about 7,000, median 7,000 to 8,000 cells being sequenced, which is pretty good for single cell DNA sequencing. And I'm pretty sure a company is working on to improve this capture rate to 10% or 20%. Allele dropout that was estimated from the common SNP loci was about median 8.7%, but it never really exceeded 20%. And this is the per amplicon performance per cell, and we got about 24x coverage per amplicon. And some of the amplicons, which covers mostly the GC-rich region, suffered low coverage, but these problems were overcome by using the HiSeq and also NovaSeq machines. So when we used the MiSeq for this project, we had low coverage for these amplicons, but by increasing the sequence depths, we were able to overcome those problems. This is a concordance of variant allele frequency from the bulk sequencing and our pseudo bulk VAF that was estimated from the single cell DNA-seq. There's a very nice concordance, and when we separate that with the single nucleotide variants versus small indels, single nucleotide variant has excellent concordance of bulk VAF and pseudo bulk VAF. Small indel has a little more poor performance, and I think this is because the bulk sequencing algorithm, which we use a pindel to call the VAF, is really actually not good to call the VAF. So I actually trust the VAF from the single cell sequencing on the indel part more than the bulk-seq. We also try to analyze the limit of detection or sensitivity of this assay, and Mission Bio is telling us about 0.1% as a limit of detection based on the dilution assay. And I think our assay analysis also agrees to this, that in some cases where we detected the mutation by the Tapestri, but was not detected by the bulk, we were able to validate those mutations by the ddPCR. And that was ranging from 0.1 to 0.3% at the limitation. So I think the limitation of detection or sensitivity is ranging from 0.1 to 0.3% at the cellular population level. And we get this type of picture for each case where each single cell is organized in the column, and each mutation is listed as a row. you cannot see the partition for each single cell here because it's so smashed here. And the scale is here represented by 100 cells with this. So there's about 8,000 cells here listed, which is kind of smashed. And we clearly see that in each single cell, these 3 mutations, which are the most commonly mutated cells in AML, co-occur at the single cell level. And in this case number 2, these 3 mutations also co-occur and creates this chunk of foundation clones. Interesting finding starts to emerge when we look these mutually exclusive pattern. So for this case number 3, where this case had a KRAS mutation and NRAS mutation, we clearly see that this KRAS mutation and NRAS mutation do not co-occur each other at the single cell level. And since now we have 8,000 or 7,000 cells being interrogated, we are able to analyze this in a statistical way to give more confidence that these 2 mutations doesn't co-occur at the statistical level. And so by the Fisher-Exact test, really these 2 mutations did not co-occur. And similar findings was observed in this case number 4, where this case had NRAS mutation and PTPN11 mutation. PTPN11 is a downstream of NRAS, so they share the same common molecular pathway, and these mutations did not co-occur in the same cell in these cases. We kept observing this similar phenomenon for this case where IDH1 and IDH2 mutations were detected. Again, these 2 mutations did not exist in the same cell. Same here, FLT3, D835Y mutation, as well as internal tandem duplication mutation. Again, these are sparing each other. So we saw this multiple recurrent theme of functionally redundant oncogene did not co-occur at the same cellular level. And this has been widely hypothesized and explored in the cellular biology, where if you put 2 similar oncogenes into one cell, that may create a senescence, or they are actually even toxic to the cancer cells, so it may not really co-occur. And end results of these analysis was that these circus plot where we are now visualizing the cell level co-occurrence of these mutations. We previously created these types of things for the patient level, but now we are able to create the cell level co-occurrence of each driver genes, and which resulted in this heat map, which consists of about 600 cells, AML cells from these 70 AML patient samples. And each partition is a patient, but within that patient there are thousands of cells smashed and creates cellular level co-occurrence and mutual exclusive landscape. And using these information, we can also infer how these AML was developed or evolved over time and created this clonal architecture. We collaborated in a group in Switzerland to use this site algorithm to infer the clonal evolution model. And most of the cases we analyzed ended up having this type of linear acquisition model, but some cases showed this divergent structure where after acquiring one mutation, there was a separate independent branch acquiring several mutations. This case really represents this divergent and convergent evolution pattern, which is really fascinating. But after acquiring this NPM1 mutation, this clone decided to separate into 2 different branches, but each branch acquired IDH1 and IDH2 mutation independently. And again, these 2 branches independently started to acquire KRAS, PTPN11, NRAS, and another NRAS mutation. So as a result, this tumor had 6 independent clones, but all these individual clones had some sort of shared functionality because they are combined with this NPM1 alteration or IDH alteration plus RAS alteration. So this really resembled this divergent evolution and convergent evolution that is observed in our biology too, which really supports that cancer cell also has this Darwinian selection mechanism to create their clonal architecture. We also started to analyze the longitudinal samples, and we accumulated about 10 or 20 cases now. And this case really represents this longitudinal power of longitudinal analysis. This was a 74-year-old male with therapy-related AML, presented with initially the 62% blast in the bone marrow. And this patient was treated with azacitidine and sorafenib, which is a FLT3 inhibitor. and achieved complete remission and enjoyed that for about 9 months. But eventually relapsed with 75% blast 9 months later. And we had a sample at pre-treatment and the relapse being sequenced by the Tapestri. And at the time of pre-treatment, this sample had a 4 independent clone, which was the FLT3-ITD, FLT3-D835E, and FLT3-D835Y, which is very tiny here, and KRAS G13D mutation. And although this sorafenib and azacitidine made this patient into remission for 9 months, the eventual relapse clone was full of this FLT3-D835Y clone. And it really made sense because Catherine Smith at UCSF previously created this kind of a nomogram for FLT3 inhibitor and its sensitivity to differential FLT3 mutations. And FLT3-D835Y clone was shown to be resistant to sorafenib, while D835E clone or ITD clone was shown to be sensitive. So it really made sense with this case that sorafenib were able to take care of this ITD clone and 835E clone. However, this very tiny D835Y clone that existed at the time of pretreatment survived this treatment and constituted the relapse. So by analyzing these longitudinal samples by single cell sequencing really created this deterministic view of how each clone independently adapted to the therapeutic pressure of sorafenib and azacitidine. We also saw a lot of multiple cases like similar trend. This case actually did not respond to the treatment, which was a cytarabine and quizartinib, another FLT3 inhibitor. But at least when we analyze these clone-based behavior, FLT3-ITD clone was cleaned up by the quizartinib, while this did not take care of any other clones, which made sense why this patient did not respond at all to this inhibitor. So we are building a lot of these longitudinal cases now to understand the individual clonal behavior to the specific therapeutic pressure. So just to summarize, this Tapestri platform really determined the clonal architecture of AML at the single cell resolution. And this is really different from inference from the next-gen bulk sequencing, that we are really determining the architecture of clonal structure in these samples. And we revealed the cell-level mutual exclusivity of functional redundant mutation, and this enabled us to create a phylogeny tree which told us how these AML evolved and created these tumors. And we also learned the lessons about technical limitation of the platform, such as the allele dropout and low coverage of some amplicons, and also some uncertainties that is inevitable from the single cell sequencing that creates some missing genotypes, confounding the results. And I think we are still underestimating the true clonal heterogeneity because we are only targeting 19 recurrent genes so far. And we really want to expand the coverage of genome while maintaining the number of cells being analyzed. And our wish list is always to integrate the transcriptome and also proteome with the single cell, which recently there is multiple paper coming which combining the transcriptome and the genotyping. So it's really exciting time. And I would like to really thank my boss, Andy Futrell, and also Kiyomi and Feng, who really did the most of the work at the bench and also in the computer too. And also Mission Bio has provided a lot of support for our initial research, so I really wanted to thank Mission Bio folks too. Thank you very much.